Yeah. So the Hardy-Weinberg equilibrium is an equation to show that if there is absolutely no change whatsoever, no natural selection, no genetic drift, no gene flow, no mutations, no nothing, then allele frequencies in the population will remain stable, given some uh, caveats like the population is large, like all mating is completely random. So it's completely unrealistic, but it does show mathematically that given those, the allele frequencies will stay the same and the population will change. It won't change at all. It'll look the same, pretty much the same kind of variation from one generation to the next. So we're looking at flowers, flowering plants, and figuring out the frequencies. And I'd like to skip to this slide, which shows, again, it's the same as the other one, just a little bit different in its format. That shows red flowers, which could either be homozygous dominant or heterozygous, and the recessive flowers, which have to be, of course, as we know, homozygous recessive. Otherwise, they wouldn't show that phenotype of white. And there were 500 flowers to start off with, so we can get the frequency from that. 320, big R, that's the genotype, 160, big R, little r, that's the genotype, and 20, little r, that's the genotype frequency. There's a difference between genotype frequency and allele frequency. So now we're going to tease out the individual alleles and find their frequency. So we've got two big R alleles in the homozygous dominant. And in that case, we have 640 alleles of the big R's. And from the heterozygous situation, we've got 160 big R alleles. And we add them up to get 800. And since there are 500 flowers, but 1,000 alleles, because each gene has 2 alleles, we divide by 1,000, and that's how we got our 80%. And similarly, with the recessive alleles, we multiply by 2 for the homozygous individual, by 1 for the heterozygous individual. We ended up with 200 divided by 1,000, and 0.2 is our frequency. So you could also, of course, say 20% and 80%, but we did figure out by doing the Hardy-Weinberg problems that it's better to stick with proportions. Yeah, so our frequency of big R, the symbol is a little p, indicating the frequency, is 0.8. And Q's symbol we use for the frequency of the recessive allele, little r, which was 20. So now if we do our Punnett square, so this is what I was trying to do by hand and failed miserably last time. But actually we got quite a bit of it. It's just that I think we tried to get to the final result in a kind of an odd way. But if we do our Punnett square and we're looking at a population of individuals breeding now, randomly with all other members of the population, and we know that there will be 20% of little r alleles in that population and 80% of big R alleles in that population. So it's similar to doing a monohybrid cross of heterozygous individuals. And we know that in the population of the females, we're also going to have this proportion of the two alleles. So then, as in a Punnett square, that the way we did it before, we just did it with individuals. So there was a 50-50 probability in that case of having one allele or the other. But in this case, we're looking at the entire generation. But we can use the same principle of multiplying the probability. So we multiply 0.2 by 0.8, and we get 0.16. So that's our probability of getting this heterozygous individual, or individuals, I should say. The same thing if we do it from the ova and the sperm reversed. If we multiply the probabilities of big R alleles, 0.8 and 0.8, we get 0.64. And if we multiply the probabilities of little r, which are 0.2 times 0.2, 0.04. So those were our result. And so in order to get them, to get our little r, we squared these two terms, q squared. So that's where q squared comes from in the equation that I gave you for the Hardy-Weinberg. And of course, for big R, we multiplied 0.8 times 0.8 together. That's squared 0.8. So that gives us our little p squared. And then for the heterozygous individuals, we add, because the probability is getting one or the other. So you add the probabilities together to PQ, gives us 0.32. So that gives us an equation for our genotype frequencies. And our allele frequencies are 80% and 20%. And these will equal 1, and this will equal 1. So last time, I handed out the Hardy-Weinberg problem set. So give that a try, and you know you could do the first two is fine. If you do more than that, that's fantastic, but you could just do the first two, and I've sent out the answer key, I think. I'll send it out to the whole class today after we finish. So this is all assuming there is no change due to natural selection or any of the other causes of evolution. And so far, that's all we've really talked about, is natural selection being a mechanism for evolution. So if we were to define natural selection, we would say natural selection is the selection of certain alleles over others in the population because they confer traits that are adaptive for survival. So that's natural selection, which is just a mechanism for evolution. We know evolution does occur within populations. If we're looking just within a species population, it's called microevolution. Just changes in gene pools. Gene pools are all the genes in a population from one generation to the next generation. It's quite gradual. The causes of microevolution are, sure, yeah, natural selection, which we've talked about, but also a phenomenon known as genetic drift, which I'll get into next, and gene flow and mutation. Genetic drift is the alteration of the gene pool of a small population due to chance. It's just purely incidental, pure chance. A couple of things cause it. One is known as the bottleneck effect, and that reduces genetic variability, usually following some large disturbance that removes a large portion of the population. So the surviving population doesn't have the allele frequency of the original population because a lot of individuals disappeared, taking their alleles with them. So the frequency will not be the same as the original population. So the bottleneck effect, and I'll show you that in a diagram in a sec. The founder effect also may lead to reduced variability when a few individuals from a large population colonize an isolated habitat. And there's an interesting human example of that. I think we talked about it. It could have been in my other class, but that is a population of, I believe, Mennonite or Amish. Anyway, it was Philadelphia. And the founding population was 3 couples only, 3 couples. And so, and for a while, there wasn't any new input of genetic material. And so some recessive and even dominant disorders became more prominent in the population because they were concentrated in a small gene pool, which is interesting. So this is a diagram showing genetic drift. Here is generation number 1. And the frequency of the dominant allele is 0.7 here. You could just count them. The reds are 0.7. 3, 4. Well, they're supposed to be 0.7. The frequency of, oh yeah, they are because of the heterozygotes. The frequency of little r is 0.3. But for whatever reason, possibly insects didn't visit these particular flowers. Or if it's wind pollination, some pollen just simply didn't land on those flowers. So they didn't make it to the next generation. Only 5 of them did. Only 5 left offspring. So in generation 2, the frequency is different. Little p is now just 0.5, and little q is also 0.5. And then, oh my goodness, in the next generation, pollination was really sparse. Only 2 individuals left offspring. Well, guess what? Both those individuals were homozygous dominant for red. So what is there left? The red allele in this case. So little p is 1, little q is now 0. There's no white alleles left in the domination in the population. And we call that a fixed allele. So here now R is fixed. It's fixed. There are no white alleles left. So that's just a chance event. Those individuals just did not happen to be pollinated through really no fault of their own. The bottleneck effect is such in a diagram. So you can see that here there are different alleles represented by yellow alleles, blue alleles. And the population got really small at some point. And then some of the alleles were lost. So our yellow alleles are lost. The surviving population only has these alleles. So that is a real reduction in allele variation in the population. And there's lots of examples of that. I'll just give you one for now. This is a cheetah. And the disturbance that caused the bottleneck effect for cheetahs is hunting. So there ended up being a very small population of cheetahs. And so there was a small gene pool, and it became very concentrated and not a lot of variation, which leaves cheetahs a bit more vulnerable to disease and that kind of thing because there isn't all that much variation. So this is an example of the bottleneck effect. And I read in a paper that the cheetahs are so closely related that you can graft the skin from one cheetah to another cheetah and the immune system would not reject it. So they're very, very closely related individuals. And a relatively small gene pool. So I don't have an example here of the founder effect, but in British Columbia, we've actually experienced the founder effect in all of our rivers and streams because British Columbia was covered by ice. I'll just write it up here. founder effect. So it's different. This here is the bottleneck effect with the cheetah. So the founder effect is different. The founder effect is when a small group branches off, migrates away or something from the parent group or population and colonizes a remote area. So that means that that small group has taken with them a subset of alleles. And so that small population with that different subset of alleles will evolve differently than the original population. In the case of British Columbia, when the province was covered in ice, well, that was it for any freshwater organisms that lived here, except for two refuges, North Vancouver Island and in the interior. But when the ice melted, where did all our fish come from? Interestingly, some came from the ocean. So with the rebound of the continent, like the ice is really heavy and it presses down on the continent by the water's edge. So when the ice melts, the continents rise back up again. It's called isostatic rebound, traps some water along with some saltwater fish. But that water isn't going to stay salt water for a long time. It's going to eventually become fresh water, and only the ones that can adjust to fresh water will survive. So we've had a few species from that experience. And in particular, of course, the most famous one is the three-spined stickleback. So not only is the population isolated, but has new ecological opportunities. So with new ecological opportunities. And I'm going to talk about that a lot more when we talk about speciation. And other areas from whence came our local freshwater species are, interestingly, the Mississippi area. Because when the country was glaciated, when it melted, there were water bodies, like Alberta was under a great big lake, pretty much, not completely, but pretty much. And so water changed direction quite a bit. And so there just ended up being, because of all the changes in water direction and rivers flowing and that kind of thing, there just ended up being some species from the Mississippi here. And really, we have quite a scant number of freshwater species in BC because we were under ice 15,000 years ago. So I think the last time I checked the guide, I think there was 57 freshwater species of fish. It isn't really that many, considering the size of the province. So yeah, that was the founder effect and the bottleneck effect. Natural selection. We've talked about that. Differential success and reproduction based on heritable traits. This is actually quite important. If the traits aren't heritable, well, the offspring won't inherit them. So there won't be any change. But if they're heritable, then selected alleles will be passed on to relatively more offspring. That's Darwinian inheritance. That's also known as fitness. So if you as an individual have more offspring than your neighbor, you're more fit in Darwinian fitness. This natural selection is the only agent that results in adaptation because those adaptations were selected for by the environment. So say salt water to fresh water. Only those individuals that can adapt to fresh water will survive. Another mechanism of evolution is gene flow. It's genetic exchange due to the migration of fertile individuals or gametes between populations. So that's immigration and emigration. And of course, that happens with loads of species. Birds in particular migrate and often migrate to different areas with other individuals of another population. So, and sometimes it happens that they mix and interbreed. So I thought it was interesting to look at humans. So of course we're all the same species, but there are differences in our physiology and some physical traits. They're very what's the word, like what's the word for a like a surface difference, a very small difference. You know, the differences between humans are really almost insignificantly tiny. I think it's 0.01%. So you and I differ in every 1 in 3 million nucleotides in our DNA. So we're 0.1% different. We're 0.1% different than chimpanzees. So the differences between humans, Homo sapiens, very small. And I found this picture in time, it was quite a long time ago now, where somebody used a computer simulation to meld all of the features of all people around the world and thought that we might look like this. A mix of several races. Because the world is a very multicultural place. It's becoming more and more so, of course. So I just thought that was interesting and a possible effect of gene flow. Okay, and let's talk about the ultimate cause of variation. So no alleles will change until they're mutated. So you won't get any new, different, unique alleles in a population unless an allele mutates. So it's a change in DNA represented by changing alleles. So when what DNA is, I mean, I'm going to do this in 1200, but just to tell you now, is a sequence. You probably know this already anyway. A sequence of nucleotides, C, G, T, A, and of course it's double-stranded. And the strands bond, the nucleotides bond with hydrogen bonds, always T with A and C with G. So that could be a sequence. It'll be much longer than this, of course, that contributes to a protein. So it codes for a protein. This is actually a very interesting topic at the moment because I wonder if I'll have time to talk about this. I'd love to. Because the vaccines that are coming out this year and early next year, they are RNA vaccines. So it's interesting to know a little bit about DNA right now. Anyhow, maybe I'll get to that and maybe talk about that a bit later. So that's what DNA is. So this sequence is an allele or a gene. And what we mean by mutations is that when it's replicated or even before it's replicated, this might disappear altogether due to radiation, UV radiation, or due to chemicals, or due to some anomaly in meiosis. That mutation will only be transmitted in gametes. So if a mutation, for example, happens to an organism say we're talking about animals on their skin, then that won't be transmitted. It's only if the mutation occurs, pointing to my ovaries, they're down there somewhere. And the ovaries are the testes. And then there's an immediate effect on the composition of the gene pool. This is the original source of variation. The original source of variation. The mutation rate. So we know mutation occurs. You just have to look. Remember domestic dogs from wolves? The difference in domestic dogs, those are all due to mutations. So those are all mutations that occurred to the wolf's original DNA that kept mutating over time. So mutations occur at a rate of 1 over 100,000 genes per generation. I'll just talk about humans. For humans, usually mutations don't make any difference because of reasons we'll get into in 1200. So usually don't affect protein synthesis. I missed something up here. Sequence is an allele that codes for a protein. We are protein-based life forms, so we're made of protein. So mutations occur approximately 1 in 100,000 genes per generation, and there are scientists that know mutation rates, so they can even predict mutations in the future. But they usually don't affect protein synthesis. And if they do, it is usually deleterious. But sometimes it's beneficial. It's even adaptive, a little bit more beneficial than some other individuals. So mutation is the ultimate source of variation, the original source of variation. And that variation is the substrate for natural selection. If there wasn't anything to choose from, then there would be no selection. Yeah. Not all variation is heritable, so we talked about that a little bit. The environment can alter your phenotype. We didn't see the hydrangea before, but there are hydrangeas, some of which are blue and some of which are purple, not because of their genes, but because of their environment, the soil in which they are raised. There are some butterflies whose color changes are due to seasonal differences in hormones. So these butterflies, these are genetically identical. But from one season to the next, they change color. And that's very common in animals. So the lynx in the hare, if you recall, we had the lynx in the hare story. The hare that lives in the Arctic is white, right, in the winter, hides in the snow, and brown in the summer. Yeah, it's very common. Very common amongst birds as well. So variation is important. So the variation due to seasonal changes in hormones. And the hormonal changes are triggered by the climate, length of day usually. So Most variation in populations occurs as quantitative characters, things like height, for example. It's variation along a continuum um, that indicates polygenic inheritance. But a few are discrete. So a red versus a white flower color, that's a discrete trait. It's either red or it's white in this case. Polymorphism is the existence of two or more forms of a character in high frequencies in a population. And really, polymorphism applies to discrete characters, not to the ones that are on a continuum generally. So what causes this variation? Geographic variations are differences between gene pools due to differences in environmental factors. And natural selection may contribute to this geographic variation. And I'll show you how that happens in a moment. Like the fishes here in BC compared to the fishes that are in the Mississippi, they look quite different now. But we know that they're related because their DNA has been sequenced. I think I need some water, just a sec. Sorry about that. In different areas, so they get isolated. So for example, these are mice. And there's geographic variation between the isolated populations. So one population is over here, but they got separated probably by the mountains. So this is quite a mountainous region here. And the other population from the original population ended up over there. So they're isolated from each other. In other words, they have separate gene pools now. Maybe they were one population before, they got separated by the mountains. Now they're two separate populations, and they took with them their own gene pool. In this case, something odd happened. Normally, this is a house mouse. They're diploid 40. You know, you and I are diploid 46. We have 23 pairs. But in some population, one population, the chromosomes fused. So the diploid number went down. So in this population, they're no longer 2n40, they're 2n39. So that's a change in their gene pool. That's quite dramatic. And there's lots of really interesting things that happen to populations because of geography. So a cline is a geographic variation that correspond to gradual changes in the environment. So interestingly, the body size of North American birds tends to increase with increasing latitude. Why would that be? Why would they evolve differently? What is the advantage to being larger in a colder area? Remember, they are endotherms, like mammals.
More body fat?
Yes, more body fat, more body volume, less likely to lose heat. There'll be an example, I have a diagram in a moment, of a yarrow, which is a kind of plant along an altitudinal gradient. Why would these plants evolve differently? So taking a look at the diagram, this is the Sierra Nevada down in California. And at the higher elevations, the plants are quite small. At the lower elevations, the plants are quite large. So that's a cline, a gradual change in size with geography. Why might that be? Why would they be smaller at a higher elevation? What do they have less of? And what about temperature? Any
Less
differences?
water?
Sorry?
Is it less water?
Yeah, there could be less water. Yeah, because there's more drainage at a higher elevation. Quite possibly, yeah. It's also colder, less time to grow in the growing season. Some alpine regions just only have really a month or so. So yeah. So genetic variation. How do we get it? Mutation and sexual recombination generate genetic variation. So offspring look different than their parents because of that. New alleles originate only by mutation and are heritable only in gametes. There's lots of different kinds of mutations. There are insertions, deletions, switches. In stable environments, mutations result in little or no benefit or are harmful. The beneficial rare mutations, however, can occur. HIV, for example, mutates and becomes more resistant to antiviral drugs. So mutations are sometimes beneficial. The genetic differences between individuals in a population is most often due to sexual recombination of alleles. So from one generation to the next, the reason that we're all so different is because the alleles of our parents were shuffled when they mated. And there's so many combination possibilities, so many. So, and I know I've said this before, but I'm going to say it again anyway. So the probability of you getting your exact chromosomes from mom is 8 trillion. Of getting those exact 23 chromosomes that you got. And from your dad, 1 in 8 trillion. So your combination of chromosomes that you personally have, the probability of that is 1 in 64 trillion. That's how unique you are. I mean, really, there's only 7 billion people on the planet. Yeah, not even close to a trillion. So there you go. Loads of possibilities of recombination through sexual reproduction, in particular through, through meiosis. And we looked at meiosis with Mendel, segregation, the law of segregation, independent assortment. What else contributes to variety? Well, diploidy, that's having two sets, balanced polymorphism preserve variation. So let me talk about diploidy. It often hides genetic variation from selection in the form of recessive alleles. So only the dominant allele will be expressed. So the dominant alleles kind of hide recessive alleles. And balanced polymorphism is the ability of natural selection to maintain the stable frequencies of at least two phenotypes. So there's polymorphism in a population due to more than one phenotype being successful. So diploidy has a component with what's known as a heterozygote advantage. Yeah, so a heterozygote, if a heterozygote has a greater survival and reproductive success than either homozygote. So it's very odd in sickle cell anemia. If you have two dominant alleles, then you don't have sickle cell. if you have two recessive alleles, that is the sickle cell condition, it may be fatal for reproduction, but the heterozygous individual has an advantage, which is that this individual is more resistant to malaria. Than a homozygous dominant individual. It's an interesting case and very well documented. So here's the distribution of sickle cell anemia. So malaria is caused by protozoan, Plasmodium. And the frequencies are quite concentrated. Yeah. Fewer and fewer from the center. So the farther away that you get from the center of the allele concentration of the homozygous recessives, the less malaria there is, because there are more heterozygotes. Or I should say heterozygous individuals. Frequency-dependent selection. The survival of one phenotype declines if it becomes too common. It sounds weird, but let's look at this snail. This is a parasitic snail. It causes a schistosomiasis or something like that. So infections, these are four different common clones, which are snails that are infected, and they have a higher rate of infection. Why might that be? That's because they're common. So they're easily found by infectious agents. In this case, it's also a protist. So eventually, these rare clones start to appear because they're not as recognizable, and most importantly, because they are rare. So they're more difficult to infect, being rare. Yeah, neutral variation is a genetic variation that results in no competitive advantage. So an example of that might be human fingerprints. Yeah, somewhat difficult to demonstrate in nature. So let's look at natural selection. Evolutionary fitness is not direct competition, but the difference in reproductive success can be due to many variables. Darwinian fitness is the contribution of an individual to the gene pool relative to the contributions of other individuals, and relative fitness is a contribution of a genotype to the next generation compared to contributions of alternative genotypes for the same locus. So survival doesn't necessarily increase relative fitness. A relative fitness is 0, for example, for a sterile plant or animal, which won't contribute anything to the next generation. So let's look at how natural selection will change a population from one generation to the next in a phenotypic range. So there's 3 ways. There's directional selection. So selection in the direction of a particular phenotype, in this case, beak depth. So diversifying selection favors extreme over intermediate. Stabilizing favors intermediate over extreme. But I'm going to show you that in a diagram because that is the most, the best way to look at it, if you can look at them all together. So here we have the frequency of individuals having a particular phenotype. The light colored mice here are rare, medium, in the middle, dark ones are also less frequent. So directional selection moves the curve over to one end of the population's phenotype. Diversifying selection will select for the extremes. So for whatever reason, the intermediate is selected out. Stabilizing selection will select for the most common individual, the most frequent individual. Okay. I just have a couple of other things. Is that okay if I just carry on? No, it's gone on a little bit long. My lectures tend to go on a little bit long. Natural selection maintains sexual reproduction. It sounds like an odd thing, but it's because of the genetic variation that's produced during meiosis. It gives natural selection something to select. So that variation is of greatest importance to the continuation of sexual reproduction, because there are disadvantages. Yeah, you can't produce as many offspring by sexual reproduction. Yeah, but the variation that's produced by meiosis seems to outweigh that disadvantage. So sexual reproduction is probably here to stay in populations. So let's see what that looks like. Here is an asexual reproductive situation. An asexual female just makes clones. This is quite common in nature. But there is usually a phase that involves sexual reproduction. So look, each clone can have 2 of its own clones. And those clones can have 2 of their own clones. That can happen quite rapidly. But with sexual reproduction, only the female reproduces. So if the female reproduces one female and a male, which of course is not necessarily the case, but there is a 50-50 chance of being male or female, the male won't produce offspring. It will generate offspring, but won't produce offspring. So it's only the female. So that's why sexual reproduction does not produce as many individuals as asexual reproduction. So sexual selection is a type of natural selection. It's a type of natural selection that leads to differences between sexes. If sexes look different, that's known as dimorphism, and you've probably seen it in ducks and lots of other animals that look quite different from each other. The male looks quite different than the female. The type of selection that's intrasexual is direct competition between members of the same sex, same sex, for mates of the opposite sex. So males have these secondary sexual characteristics like antlers that they fight with. Intersexual selection, however, is mate choice. So one sex is quite choosy when selecting. And that gives rise to this amazingly sophisticated and secondary sexual characteristics. So we'll see what some of those are like. The lion's mane. This is a frigate bird. The male frigate takes half an hour to blow up this enormous membrane in its throat to attract the female. This is a grouse. So much color. This is a case of intrasexual selection with the antlers. And I suppose the lion is also intrasexual, isn't it? The male lions fight for females in their pride, don't they? I think they do. Yeah, so that's intra as well. Yeah. Very, very cool. And of course, the peacock. Yeah, that's intersexual selection, where females choose the most adorable male. But of course, that confers fitness. So there is a reason. Because can you imagine? I mean, just looking at this peacock, how much energy has gone into making those feathers? You know, probably most of the energy that this male peacock has. But it's worth it because that peacock's genes are likely to get into the next generation. Yeah, so it's a big deal. But one thing to remember is that natural selection does not produce perfect organisms. It's just simply by accident. There's historical constraints. Yeah, we're constrained. Our ancestors were four-legged. I don't know many people that don't get back problems at one point or another in their life. And adaptations are compromises. Humans are athletic because we have flexible limbs, but there's a compromise. They can be dislocated or torn. Um, Not all evolution is adaptive. Yeah, chance plays a huge role in evolution, and not all changes are for the best. And selection edits what already exists, existing variations. There's no pre-thought, no conscious effort. It just so happens that you get a mutation, you get an allele, it's pretty good. So it might be selected for. Yeah, new alleles don't arise as needed. So that's an important point to remember. And that's where I'd like to end. Thank you for your patience. I really